What I'm going to be talking about is the role that ARS plays in the breeding program here in the state and also across the U.S. One of the thing is that the U.S. Rice Research Program is scientists addressing the needs of the rice industry. And the major mission that we have is to conduct research to help keep the U.S. rice industry competitive in the global market by assisting in high yield, superior grain quality, pest resistance, and stress tolerance. The strategic goal we have is to increase the efficiency of the U.S. rice agricultural production system, and this is to expand, maintain, and protect the genetic resources, increase our knowledge on genomics, and biological process, and improve the efficiency, health, and the value of the national crop. This particular goal that we have is to maintain the rice industry not at the present level, but at an increased level. And the only way we're able to do that is to be able to understand the genetics and the quality issue of the rice. Major issues that we face, of course, is yield, and milling yield goes in with field yield because that is what the farmers get paid for. They don't make any more land, so you got to find out how do you grow more rice on the acres that you have. And also, if you're growing more rice and it doesn't mill very well, then you're going to wind up with reducing the amount that you get for the rice that you grow. And the other thing is called a niche market, and that is that you can grow specialty rices. Therefore, you can get paid a higher price for growing these particular rices. Everybody here is probably familiar with the Dale Bumpers National Rice Research Center, which is right next door. And it was established in 1998, but you may not be familiar with the original Rice Center, which is in Beaumont, Texas, and it was established in 1955. And this was established because they had a variety that was high yielding in 1951, but it was not a very good processing rice. So the industry suffered big from that particular era. So what it is that the rice growing states got together and says that we need to find out why this particular rice is not as good as the rice we used to have them. So they got with Bill Webb, who was a research chemist, and developed this lab for the sole purpose of analyzing rice for a superior quality. What he did find out is that rice is generally, the quality or cooking quality of rice is determined by its amylose content and its ease of cooking or its ability to be cooked. Since then, the USDA ARS has invested in 11 scientists that our sole purpose is to analyze and maintain the rice industry, and they are located both here in Stuttgart and in Beaumont. We have two individuals that are in Beaumont, and Bob Festrom came from Beaumont. He's now here in Stuttgart. They are in different areas. One of them is your molecular geneticists, and the other one is in physiology, Dave Gailey, and you have your quality, which is dealing with the cooking and processing quality of the rice. So we have a broad arrangement of scientists. As I say, rice is grown in about 5 different states. You've got Missouri, Arkansas, Mississippi, Louisiana, Texas. You got some that's grown in California, and very little bit of it is grown in Florida. But Arkansas grows about 50% of the rice acreages. What Bill Weld found out, as I said, is that amylose is a major component for the processing of rice. And what the center in Beaumont does is analyze about 10,000 samples a year from the rice breeding program in order to assess its rice quality. And they look at the amylose content, and here we see 0% amylose all the way up to about 25%. And what we generally like is somewhere between 22-23% amylose. The other thing is the ease of cooking, and that is that you have rice that is not affected if you put it in chemical, all the way down to rice that is affected and totally dissolved. And we look at its cooking properties, which is the RVA. This is one of the machines that we use to determine the amylose content. And this is the way we look at the alkaline spread. And what you do is you take the kernels and you put them in a solvent, and you let them sit there overnight and come back and look at them. If they almost dissolve, then those are very easily cooked rice. If they are not affected at all, then they become, they're your very high amylose rice. The other thing that is new that we've just started several years ago, and that is being able to look at the DNA in the rice. And we're associating the DNA with certain markers. The disadvantage up until now is that the only way that we had of determining whether rice was good or not is by vision and a very few chemical analysis. What the breeders would do is go out and look for things that are high yielding, or they'll look for them being short in maturity. And with the DNA markers now, we're able to look at the rice before it is mature. What we do is we look at a particular trait that we're interested in, for example, amylose content or alkaline spread or any diseases, and then we try to find a genetic marker that will be associated with that trait. And we will do this repetitive until we are sure that if we find this marker or don't find this marker, that particular trait is either there or absent. And then once we do that, we'll give it to the breeders and they can begin to use it in their breeding program to find varieties. This is a slide that just shows that the DNA, which is in the cells of all living things, and you decipher the particular code, and in that you're able to determine what particular thing it was developed. So you got to get the DNA out, and the way you do that is you can grow up a seedling, cut the leaves off, extract the DNA from the leaves, and then you wind up with the DNA. You can then run them on particular machines to amplify the DNA and then to analyze the particular DNA fractions. And what you then begin to see is different DNA patterns. In other words, you wind up here, if you have a particular marker that is in this range, then it will be a long grain, non-sticky, which is what we like. Or it could be in this range, which is a medium grain, and this is like Bengal, and it winds up being a little bit more stickier. Or you can wind up with a very high amylose, and this is what they use in canning products. Because if you try to use the others in a canning product, what will generally happen is it'll fall apart. Now, this is good because what you can do now is look at the rice before it matures. In the past, remember, if we were going to look at the amylose content, we had to have a grain. So you had to grow it up, collect the grains, analyze it, and then the next year you grew it out again. This way we can grow seedlings up, and you can do it in the greenhouse, so you can do it year round and gain a lot of information. Okay. Another thing that we're looking at is rice blast, which is a disease. And this is something that we did not have any chemical analysis for, but now we have been able to associate markers. And since there are different fungi out there that call different races, just because you have a resistance to one doesn't mean you have a resistance to another. It's like a cold. You can catch many different colds, and you have to be immune to them all. So we now have 5 markers that we can use, and when we run them by using the DNA, some of these will have either one type of resistance, others will have more than one. So when you cross these plants, you get the DNA out, and you can tell whether that plant is going to be resistant or is not going to be resistant. In the past, you had to grow it up, spray it, or find out if it was resistance. So in all, up until now, the breeding program generally took anywhere from 10 to 12 years to develop a variety. But because of the input that the USDA has put into it, that time span has come down to 7 to 8 years in releasing a variety. And the main reason is because we are able to now get into and look at the rice before it's matured, right after crossing, and we can determine whether or not that rice has the particular gene for becoming a good quality rice, or whether it does not have that particular gene.